So what I want to talk about, actually, let me sum this up a little bit. So you can't inherit a tumor, but you can inherit increased cancer risk. Okay. And there are many examples. What I want to talk about next is that the world is a slightly more complicated place. I've been telling you that normal cells, say you had a mutation in RAS, it's going to lead to a tumor right away. This is oversimplified. All right? And I want to explain a little bit about that so that you know more when because all of us are going to run into cancer in our lives, usually, hopefully not in a personally tragic way, but oftentimes in a tragic way. So what I want to show you is a picture which is in today's handout. If you turn to the next page of today's information-packed handout, I want to show you this really monster diagram, which looks sort of like an extremely complicated subway map. And clearly, I will not make you responsible for this. However, this is actually a 12-year-old figure, and for sure it's gotten more complicated than this. It's just that this is a really nice, pretty picture. By now, it's probably twice as complicated. But if you look, growth factors, that's the receptor, R for receptor. That activated RAS. Okay, maybe not directly, but close. And then RAS does a whole bunch of things going into this, which is the nucleus, changes in gene expression. And deep down here somewhere, near the bottom, I think, is P53. So I wasn't lying when I said P53 and RAS are important players. It's just that they're one of many, hundreds of players. And a couple of things to think about is that means every single one of these is a target for mutation that could potentially lead to cancer, which is also why cancer is such a leading killer of people, because there's so many different targets out here. So the real thing is more complicated than this, and it's gotten more complicated even than this figure in the 12 years since that figure was put out. But this just shows you where those things are in the big picture. And those are all genes that control cell division, which are then possible things that could be involved in cancer. Another complication, so it's not just as simple as those few genes, although those are important. It's really, actually, let me go back. Better to put it here. It's really, you have a normal cell, and that many mutations lead to a tumor. This is what we call the multi-hit model. And it also explains why it takes so long for cancer to occur. So, for example, in colon cancer, which has been well studied, figure 1825 in Campbell, if you want to see some pictures of this. If you have a normal colon cell, what typically happens first, but not always, is you end up with mutations, both copies of a gene called APC. If you mutate both copies of APC, you end up with a small growth, which is sometimes called a polyp. So the idea is that why APC is first is not clear. Maybe it's a hotspot for mutation. There are some of those in cells. But typically, so normal colon cells, if they get mutations in both copies of APC, so that's a tumor suppressor gene, those cells start to grow slightly faster. And that polyp is a benign tumor. That's what they do colonoscopies for, and they remove those things. If it's not removed, there's a chance that then they can pick up a mutation in RAS, and you end up with a benign tumor, a more significant polyp. Then if there are mutations in more genes, particular ones, you end up with a malignant tumor. And the thing about this that makes it sort of insidious is that along this way, the cells divide faster. Therefore, mutations are more likely. So, a couple of things to know. This progression takes many years, and it's not guaranteed. One of the huge mysteries in cancer treatment is what fraction of those nasty little polyps or little tumors or lumps in people's breasts or lumps on people's prostates are going to end up bad, and what fraction are just it would never do you any harm. And we don't have a good way at this point to tell them one from the other. We can know some levels of that, but we can't really predict in many cases. So there is no guarantee that a given tumor is going to progress down this way, but they sometimes do. And once they start down this road, it's like a ball rolling downhill. They pick up speed. And why is that? When you mutate the APC gene, those cells are growing faster. Cells which divide faster, if you remember, mutations happen when you copy your DNA before you fix the damage to it. So if you're in a hurry to replicate, you're more likely to replicate before you fix the damage. So the faster you divide, the more chances are for mutations to happen. Also, the faster you divide, sometimes it can be so fast that the chromosomes actually get messed up and broken. So by the time you get down the end, the DNA in the cell is quite abnormal. And so what happens is all along this way, the mutation rate picks up and up and up, so you pick up more mutations, so it divides faster, so you pick up more mutations, so it divides faster. So typically, these things start slow, and then they pick up speed, which is why screening for polyps is good, because if you snip that guy out, you know it's not going to get any worse. Right? But they can, although they do not always. And that's also why it takes such a long time, why cancer is typically a disease of old age, because it takes a long time for these mutations to accumulate. And that's typically why it doesn't happen until you're much older. Questions about this progression? And they're starting to work them out for many other cancers to try to figure out what's mutated, and also to be able to differentiate things that are going to cause you trouble from things that aren't. That's sort of one of the frontiers of research, and they don't have good answers to that, but you'd like to know. All right.